while we wait for Peter, I'm here to tell you that we're going to listen to very interesting speeches today for this first HortiQD webinar on commercialization history of quantum dots in optoelectronics and bioimaging sensing. So we will be listening to Professor Nico Hildebrand. Thank you for joining us. Also for Dr. Kim Donolf, which you can actually already see. And then we will have Dr. Peter Rice. And then in the end, that's an important thing, we will have Q&A session. So please ask your questions. We will collect them, and at the end of the entire webinar, we will be asking your questions to the speakers. So I don't know if Peter, can you now use your microphone?
I hope so. Do you hear me?
Yes,
yes.
We can hear you. So if you want to add something to my introduction, please do it, and then we will proceed if you agree.
It's perfect. So it's I had some connection problems, but these are now solved, and so I'm really happy to have everybody here, and I'm looking forward to all the talks in this first HortiQD webinar.
All right, so I think we can give the word to Professor Hildebrand, that he will tell us about quantum dots for simple and sensitive multiplexed biosensing. So please, Nico, go along, go forth.
Absolutely, thank you. So let me share this, and let me see if I can put the laser pointer on. Wait, now it's moving. Can you see anything?
Yes.
Okay, so you should see my NHL logo and quantum dots and pictures.
Yes.
If that's what you see, then it's good. Okay. Well, welcome everybody, and thanks for the invitation to the webinar. Happy that you made it, Peter. I guess the CEA restrictions make problems for connecting. I will talk today about quantum dots for biosensing. Not so much for commercialization, though, because there's not a lot of commercialized biosensing products with quantum dots, apart from that you can buy them and use them in your research. But I hope it will be interesting anyhow. I'll give a short introduction of myself in the beginning. Yes, here it worked. Do you see the second slide with yeah? Good,
Sure.
then everything works. So like Peter, I'm originally German, so I grew up here in the north of Germany, between Hamburg and Lübeck, and then studied. And here on the right side, you always see some pictures for tourist information if you ever want to go to Bad Segeberg. It's not a bad city. Bad just means bath in German, so it's called Bad Segeberg. Then I moved for studying to Berlin and did my PhD in Potsdam, and then moved over to France. So like Peter, I'm also a German and French citizen. So we both maybe kept our original citizenship and took another one, and maybe I'm getting a third one now from Canada. Let's see. So here to Orsay, and then I moved to Rouen. And after that, I lived for 2 years in Asia and South Korea, in Seoul, where I was a guest or visiting researcher in the lab of Jwami Nam, who's doing plasmonic nanoparticles. And after these 2 years in Asia, I moved in 2023, so 1.5 years ago, more or less, to Hamilton. So yeah, here it's actually moved a bit down. So you see, well, now I can say, the unity between Canada and US is still strong, because my yellow ring here is half on the Canadian Lake Erie side and half on the American Lake Erie side. But actually, it moved down. It should be here in Hamilton. So the Niagara Falls are here, Toronto is here, and Hamilton, where McMaster University is here. Hamilton is the city of waterfalls, so they claim to have the most waterfalls in the world. Check it out. I haven't seen so many yet, but it's just because I haven't been out to the waterfalls. Okay. You also might know me as Nico at the Sword of Light. This is if you have Amazon, this is a series, and it fits very well to me. Just that they got the hair wrong. I have hair on the side and not on here, but this Nico has hair in the center but not on the side. But it's a superhero with a Sword of Light, and that's I like that because that's also what we're doing with lights and quantum dots. And I'm also known as Captain Fred, so this is me and my team in action here. This is the actual real Captain Fred. This is Theodor Förster in the 40s. And don't worry, I'm not only showing photos about me and others, I will also talk about quantum dots in a little while. So this is the real Captain Fred, and I will tell you what Fred is because I will talk about quantum dots and Fred. So this is Theodor Förster. I like this photo very much because, you know, in the 40s, you could still smoke the pipe in the lab while you're making experiments, as you can see here. Good, quantum dots. Finally. So as many of you probably know, quantum dots finally got a Nobel Prize in 2023, not for biosensing. And so we worked with quantum dots for multiplexing because they're colorful. And on top here, this is a conference that Peter also co-organized in Paris. And I always show this to tell to the young people, if you're at a conference and they're taking the photo, always make sure you see the camera because you see here are the three Nobel laureates. They were all there at this meeting, and I'm also here. You see in this ring down there, but you can't see me. I just know that I'm there because here is another colleague who's talking to me. So, you know, I could say I was probably there with the three Nobel laureates, but no proof apart from that I'm telling you. So if you're at the conference, always look at the camera. Um, so the quantum dots got the Nobel Prize, but not for biosensing, right? Although they have been proposed for this very early on, and in the beginning it was a kind of hype that people said quantum dots will replace every other dye and things that you have, but they didn't. Like nothing replaces nothing anyway. You know, there's always some aspects of sensing or of any other application where you will use one or the other. And quantum dots have specific benefits and advantages, but dyes also have. But then it's also very polarized, I would say, discussion about the toxicity of quantum dot. Quantum dots are always considered as very toxic, but in fact, everything is toxic, right? Toxicity is always a problem of concentration. So the more you put, the more toxic it is, right? So that's what they say. The solution of pollution is dilution, because if you dilute things very much, then it doesn't exist anymore. And there are studies where they actually compared dyes like DAPI. Nobody cares about the toxicity of DAPI, right? Everybody's using it, but DAPI has the same toxicity as cadmium selenide quantum dots that are well protected, depending on how you use it. And so the message here is that toxicity actually needs to be properly assessed and not simply assumed. Another example is phototoxicity. So I also work a little bit with upconversion nanoparticles, and nobody seems to have a problem shining 6-watt lasers at cells because they're in the NIR. But this is so strong in intensity that it even heats the water. But it's just NIR, and people assume, well, NIR is deep penetration, and it's and they say, well, UV is. So what I hear often, UV is dangerous for cells, and NIR is not. But that really depends on the experimental setup, on the situation. So an interesting paper is this one here by my colleague Igor Medins from NRL, where they did a meta-analysis of cellular toxicity for cadmium-containing quantum dots, and they looked at 307 publications and had over 1,700 cell viability-related samples, and to see how quantum dots are really toxic under which situation. And they found that toxicity is closely related to the surface properties, so what is on top of the quantum dots, how they are protected, also the diameter, the assay type, and the exposure time. So there's no way saying quantum dots are simply toxic. You need to see it in the context of the measurement. Okay, quantum dots. Short introduction about why we like them, or why everybody likes them, because you can produce quantum dots that emit in any color between the blue or UV up to the NIR, depending on the size that you're using it for. So here in the center, you see quantum dots. They're all cadmium selenide. The small ones are blue and the large ones are red due to quantum confinement. They have these very narrow and Gaussian or Lorentzian, if you're a fan of Lorentz, a Lorentzian-shaped emission. And depending on which semiconductor material we use, we can tune them very, very broadly. So they're also excitable at any wavelength. That is also important because they absorb any light below their emission wavelength. And they have also very large surfaces, which is nice because we can put a lot of biomolecules on these surfaces. And they're very bright and photostable. And you can use them in many applications in FRET biosensing or normal biosensing. You can also use them in arts and technology. So this up here is a TM that a student in my lab when I was still in France did. He was an Italian student visiting Paris, and most of the time he was in the museums. That's what you should do if you're in Paris. And so he found that La Danse from Matisse really resembled his TM, and I kind of agree. And here on the right side on the bottom, there's a piece of art from Peter's group from 2014. And well, here's the QLED, so where quantum dots really got into technology for the screens that you see in the if you go to the shopping mall. So our motivation for working with quantum dots is, and I drew this myself, so you can make your own opinion about it if you like my drawings or not. So we're exposed to many things, and I only drew some of here on the left side, but some of us are smart, and so if we use light and molecular and nanomaterial interactions, we can improve the resolution and the sensitivity of biosensing. And if we can do that, we'll get a better diagnosis. If we have a better diagnosis, and that might be in the environment or in a body or wherever, we can have a better understanding of what's happening and then maybe have a better therapy for the human or a better therapy for just polluting less. So all of this, like for a lot of biological or medical research, would not be necessary if we would just live smarter, right, and not pollute, and we would all get less cancers and stuff like this. But this is probably hopeless and will always happen. So, well, it makes us researchers have some work that's also good. So we like to use energy transfer to quantum dots. And for that, we work with lanthanides, and I will talk about this. And we can make energy transfer from lanthanides to quantum dots, from lanthanides to fluorescent proteins, from lanthanides to quantum dots to dyes fluorescent proteins. And these other materials also had Nobel Prizes even long before the quantum dots. So, which actually means I'm working in a field where there's no more Nobel Prize to get. But anyway, people like me don't get them anyway. So what is FRET? FRET is first resonance energy transfer. You can also say fluorescence resonance energy transfer. And what you need is a donor that emits, so for example, a quantum dot, and an acceptor that absorbs, could also be a quantum dot, and they need to be close together in a range of 1 to 10 nanometers or so. And then you can measure this distance. So this E FRET here is the FRET efficiency between them. And for that, you need to make two measurements. First, the donor in the presence of the acceptor, and then the donor alone. And then you take their intensities or the lifetimes, and then you can calculate this FRET efficiency. And you can use this for different biosensing applications. So these are the lanthanides we're using. These are 3 examples. We're working a lot with this one in the center, which is terbium complex from the company Lumifor in California. And so you can see that the emission of the terbium has these different green lines here. So if you're familiar with lanthanides, this will not shock you, but if you work rather with dyes, you would think these are 1, 2, 3, 4, 5, 6, 7 dyes. No, all these emission lines, narrow and well separated, come from this terbium ion. And the absorption is actually from the ligand. So the ligand around here or here absorbs the light, and this is in the UV, and then it transfers the energy to this terbium. And then from there you have the emission. And apart from these narrow lines that you can see here and well separated, you have this very long excited state lifetime. So if you work with quantum dots, you might know they have an excited state lifetime of a few nanoseconds. These lanthanide complexes have a very long phosphorescence in the millisecond range. And we exploit that for our energy transfer. So the first exploitation we use is this broad emission. You see here is the terbium emission in black. And if you remember from 2 slides ago, I told you for FRET, the emission of the donor needs to overlap with the absorption of the acceptor. You can see that here for dyes, so we can make it overlap with 5 different dyes. But as the absorption of quantum dots that you can see here for 5 different quantum dots, it's much broader and much stronger. So it's tenfold higher than the dyes here on the y-axis. We have a much broader overlap. That means we can make energy transfer over larger distances if we use quantum dots as acceptors and terbium or europium or other lanthanides as donors. Now, 5 quantum dots or 5 dyes can quench 1 terbium, but how do we know which dye or which quantum dot quenched it? Well, for this, we need to measure the emission. And here comes the second advantage of quantum dots. So if you look at the left side, the dyes, they all have these second shoulders. So on these shaded lines that you see in the background, these are filters. And if we're measuring with the orange filter, well, the orange dye has the highest intensity there, but you can see that the green and the blue dye also have a large contribution to there. And if we use these narrow quantum dots that you can see here on the right side, there is almost exclusively emission from the orange quantum dots, so we can make better multiplexing. And the assays that we're using, and I'm going to show one example for imaging actually in vivo, but the principle is the same. These assays that we're using have been developed a long time ago, not with quantum dots, but with dyes. Usually they are called sandwich assays because you have 2 antibodies that you can see here, one with a terbium and the other one with a dye or the quantum dot. And if you excite them in the UV, you have this very long emission of the terbium, this very short emission of the quantum dot, and you also have a lot of autofluorescence background from your biological sample. So if you add the biomarker, or if you add your kit to the biomarker, you will have this binding of the antibodies. That's why it's called a sandwich assay. And then they're close together and we can have FRET. And FRET means that it's the green emission from the terbium is quenched. But what is more important is that the quantum dot or the dye now gets the energy from this long-lived terbium and also becomes long-lived. So you can see this orange here on the right side is all new, and it's only there if the biomarker is there. So now if we use time gating, so we excite with a flash lamp or with a laser, and we measure the intensity after a certain time, then we will have 0 background and can quite sensitively measure this biomarker, which could be really anything in clinical diagnostics or a marker in environmental and water sensing, for example. So it doesn't require, as you see here, any separation steps because the FRET gives us a unique signal. So only when it's combined, and that means that we don't need any incubation and separation as you would have for an ELISA assay, for example. There's no magic on it, so you can use this technique, time-gated FRET, which is called TR-FRET or TRF or HTRF. There are other brand names on different readers. I have shown only some here on the bottom. We also use this clinical plate reader from Thermo Fisher Scientific. We developed our own instruments with Edinburgh instruments together, and we also do this with a time-gated microscope where we just do time gating. So it can basically be done in any lab. If you have this instrument, you don't need to custom build your setup. And then we can do color multiplexing in this biosensing. So, well, as I showed before on the spectra, you excite 1 terbium, and then you can make energy transfer to different colored quantum dots to measure 3 different DNAs. And you might say, well, the 3 DNAs here are all the same. Yes, that is just because I am not good in drawing. So this is these are the same DNAs, but you have to imagine they're different. And we can also do this on the time scale, because as the energy transfer efficiency depends on the distance, the closer the distance, the more efficient it is. So the lifetime gets shorter. And what you can see here on the bottom is the intensity gives you different concentrations of the DNA and the different lifetimes. So you can see here are different slopes, 1, 2, 3 slopes, are different distances for different DNAs. And so we can use one single FRET pair and time multiplexing to detect different targets. And if we combine it together, so let's assume we have 5 quantum dots, so with 5 colors, we combine it with 3 terbiums at 3 distances, so that means 3 lifetimes, and we dope them in polymer microparticles, for example, or larger nanoparticles, we have 3 intensities, and this would scale 3 to the power of 3 to the power of 5. This is 3 to the power of 15, and this could be 14 million or more than 14 million codes that we could create, at least on a PowerPoint slide. In the lab, for sure, it's a bit more difficult. And you also have to take into account that often in solution things are not distinct. So if we're in solution and we have 5 colors and 3 lifetimes, we can actually only measure 15 because the intensity measures our concentration. But if we measure it in imaging, it would be 5 to the power of 3, and we can measure 125 different biological interactions. So let me give you one short example of an application, how we can use these and that they can be really used in biosensing. So this is about imaging, and I will talk about in vivo imaging here, where we did some experiments in zebrafish to follow quantum dots or this red quantum dots over a long time. So zebrafish are not only great because they are transparent, so you can use them ideally for optical microscopy. It's also that they are developing pretty fast. So you have cell separation like every few minutes, and after you see here, you can already see a zebrafish larvae after 1 day, starting from the 1-cell stage. And so we inject at the 1-cell stage and then see how long we can follow without any other injection. So to see if these quantum dots are really bright. And as I will show you, you can follow them over several days without injecting new quantum dots. So they're just so bright that they separate from cell to cell, and you can still see them. So the system we're using here is a quantum dot, and attached to it is a peptide, and the peptide we're just using because the histidine tag here on the end will self-assemble to the quantum dots. So the surface of quantum dots can be very easily functionalized. And then this terbium is transferring the energy to the quantum dots. And the different colors of the quantum dots here mean that we use 3 different quantum dots, a turquoise one, an orange one, and a red one. And we can also assemble an M-cherry fluorescent protein that is expressed in the cells with a His tag intracellularly on these quantum dots. And then we also add some proteins that is just for microinjection because quantum dots are a bit sticky. If you inject them and you have some proteins on it, they get less sticky. So the idea is that we have the energy transfer from the terbium to the red, orange, and turquoise quantum dot, and maybe also to the M-cherry, to the fluorescent protein, and then the turquoise can also, there's many energy transfer steps. But the system in imaging is again the same. We excite in the first 10 microseconds, we don't measure, and then we start measuring, and all the background from the living system will be gone. And we only have our signals from terbium, M-cherry, and the quantum dots. And you can see here, we inject this on the single cell level and then measure it over several days, for example. We started with cellular imaging just to show that we can do multiplexing. So we injected all of the quantum dots into different cells and also the terbium. And then you can see we can make energy transfer from terbium that you can see here in green to the turquoise, to the orange, and to the red quantum dots. And on the bottom here, you can see this triple energy transfer where we have energy transfer from the terbium to the quantum dots to the M-cherry. And so the great thing is that here in this cell on the right, there was no M-cherry expression. So we were lucky to see this in the same window because these 3 cells had M-cherry expression. So you can see that there is energy transfer from terbium to the quantum dot, but not to the M-cherry because there is no M-cherry, whereas here there's clear energy transfer. So if we do this with zebrafish, and zebrafish are not that long, it's just that when they get older, you have to squeeze them in a kind of channel, otherwise they would squeeze around. So these are captured zebrafish larvae, if you like. So you can see with time-gated FRET, we see a nice terbium and quantum dot signal over 70 hours, and there's no background. Whereas if we do the direct excitation, we excite the quantum dots, but we also excite the terbium, but we excite a lot the background. And you can see, so this is not specific signal. This is just green background, and red background also comes there from the zebrafish, so we cannot distinguish any specific signal from background signals from autofluorescence. And we can do this over 168 hours, so that's what, 7 days divided by 24? Well, several days, and we can still see it. So we inject at the 1 cell stage. Here you have many cells already after 4 hours, and then you see we have to, after 72 hours, make an image of the head and of the tail, because we can't get them at the same time. And you can see that we still have signal. It fades a bit away in the FRET channel after 168 hours, but we can still have stable emission on this. You can see here in the control, there's really nothing in the time-gated channel that you can measure. The toxicity is not much. So, I mean, as I said, everything is a little toxic, but already the messenger RNA that you inject in the beginning to express the mCherry is a kind of toxic. So the survival rates are okay. It depends on if you use old quantum dots. So we also measure the deformation rates, because a lot of these fish survive, but they deform. You can see if you use old quantum dots that already aggregated, the toxicity is a little higher, but you can see red quantum dots and RNA is still pretty okay in these deformation rates. We can also do immunoassays with quantum dots, and we can do DNA assays with quantum dots, and we can do barcoding with quantum dots, and we can do molecular logic gates with quantum dots, and we can measure distances of molecules on quantum dots. But this is all another story. So I want to thank all our fantastic collaborators who we worked with over the years, all the fantastic funders for the money they gave us. Would like to thank all the people who are on the right side who worked on these things that I showed. This is my current group in Canada. We're just building up things, so none of the results I showed today is from here, but I hope we'll soon. Well, there's a bunch of people, so we'll have some results soon, I hope. And I'll end with this FRET train here on the bottom. So that's the most important accomplishment of the French, is they built a train that is called FRET, and this can do FRET over thousands of kilometers from Lille to Marseille. And when I moved from Germany to France, I saw we can only do FRET over 10 nanometers, and they do FRET over thousands of kilometers. So thank you for your attention, and I'm sorry that I went over time, but Peter has to talk less than in the end. Thank you.
Thank you very much, Nico. I'm just here to tell you once again that if you have any question for Professor Hildebrand, you can use the proper section, and we will answer or try to answer to all of them in the end. Now it's time for Dr. Kim Donolf. She will tell us about quantum dots for advanced display applications. So Kim, when you're ready, you can go. We can see your shared screen. Yes.
Okay, great. Can you see full screen now?
Yes.
Yeah, okay, perfect. Hello, everyone. My name is Kim Denolf. I have to say that I'm totally not used to being called a doctor, although I am, but that seems like so long ago. Today, I'm, let's say, more in my role of CEO of Custom Dot. Custom Dot is a company that makes heavy metal-free quantum dot inks for next-generation display technologies. I was invited to do this presentation here in the workshop, and I liked it a lot because it also made me think a bit about how Custom Dot came to be, but also all the history that is actually behind that. So thanks again for this invitation, and I also already want to thank Johnny Steckel for helping me so much with the presentation, with the content. Yeah, this was already mentioned, of course, in the talk of Nico, but without let's say people in the early days that did work on quantum dots, of course, the commercialization of quantum dots would also not have happened. There's such a wide history on this topic. So I try to limit it a little bit because, of course, there's not too much time for me to speak. And let's say I try to like summarize the commercialization of quantum dots in different display applications, and then make a little bit of an overview of what might still happen as well. So let's dive right in. The first part is about LCD, so that's liquid crystal displays. Liquid crystal displays are let's say the first displays at least when I was young, that were called a flat screen. So before we of course had CRT TVs. This of course has nothing to do with quantum dots. Those were those bulky kind of TVs, and that was the norm, let's say. And then LCD came along, and this was really an improvement for everyone. So what happens with liquid crystals is you produce white light in the back of the screen, and then in the front there are liquid crystals which you can control, and then these allow a certain color to pass or not. And if you have this control over these liquid crystals in every single pixel, then you can create, of course, a pixelized image, which is really nice. The LCD was already around for quite some time, and then quantum dots, let's say, became a commercial product as well, or quantum dots played a very big role in this product as well, because they actually can improve LCDs a lot. So traditional LCDs were actually made with a blue LED light that then passes through a phosphor layer, and this kind of spectrum looks a bit like this. And then when it is filtered through these liquid crystals, this is how the filters look like. You can actually create a blue, a green, and a reddish, let's say, peak. And then this is really what how every single color on a display is formed. Here we have an image of let's say the color gamut of a display. So this kind of funky diagram shows actually all the colors that can be visualized, or that the human eye can see. So this is the whole diagram, and when you select 3 colors, you can make a bit of a triangle with those 3 colors, and then you can visualize every single color within that specific triangle. So this was already very nice, let's say, but of course, as you can see, this is the phosphor here was not really pure green or pure red. It was something in the middle, more a bit something orange yellowish. So this really needed improvement. And that's why they stepped away from using phosphors as to convert to this kind of orange light. And then they used green and red quantum dots instead. And then you have this kind of spectrum here, and this, of course, can then be filtered out by the liquid crystals into 3 nice peaks. So this was already a big gain for LCDs. LCDs are very manufacturable. They are not so energy efficient because, of course, you create a lot of white light in the back. And if you want to show a black image, you actually need to block out all that light. If you want to show a red image, you need to block out part of the light. So it's not a very energy efficient display. Also, the brightness was not so good. The color volume or the color gamut was not so nice as well. And also the contrast ratio of such a display is quite low. With quantum dots, it really improved, and especially the energy efficiency, because there's less light that is lost. So light from here that is lost. Also, the brightness increased a little bit, especially the colors really improved a lot. Contrast improved a bit as well, but of course, still it was medium in contrast. It had very limited viewing angles, and yeah, you could not really do a lot with LCD in general because it's a limiting technology. So as far as the, let's say, requirements go for quantum dots in LCD, it's actually let's say, the easiest application. That's also why they came first, of course. The absorption, so the amount of light that needs to be absorbed is quite low because you have a lot of quantum dots compared to the light intensity. The photostability also didn't have to be that high because, again, there are a lot of quantum dots. There's a lot of light in the back, which was not needed to be very intense. And thermal stability would also be very low, and there's no need for patterning because you can just use a film. The first commercial LCD products actually came in 2013. QD Vision had this color IQ optic technology. Nanosys and 3M also made a quantum dot film, and this actually led to the first commercial products. So the Sony Bravia TV in 2013, and also the Amazon Kindle Fire. This was really such a major jump for quantum dots, especially if you know that they have been already made for the first time in the 70s or the 80s. So it took a quite long time, but then they were there, and they really improved, let's say, displays in general. From that initial improvement, of course, the quantum dot LCD TV is still around. You might know these kind of TVs as the QLED TVs, for example. And these, of course, also still are being improved. So before, we kind of had a backlight that was just one uniform backlight. And then the contrast in such a screen was not really that good. There was no, let's say, the brightness levels were not adjusted very well because, of course, when you filter out light, you cannot block every single piece of the light. So that means that a lot of things were not very good. And then what they actually started to do is to make those that backlight, that was one really big backlight, into a few distinct pieces. And then that allowed to locally dim a little bit, to improve the contrast and so on. But what really helped is that now actually this backlight is consisting out of mini LEDs in different zones where you can actually turn them on and off already. So, for example, if you see here this dark side of the moon, let's say, that part, of course, can already be dimmed, which then improves the contrast, the brightness, the HDR. So there is still innovation in, let's say, quantum dot LCD as well. And quantum dots, of course, still play a very big role to make this really intense and bright colors. Then a second display technology that is important here is OLED. I think most of you will know OLED. There are, let's say, two big technologies in OLED. One that is used more for smaller screens, and then one that is used more for the larger screens. There are two major, let's say, parties that are playing a role here, and that's Samsung and LG. So Samsung has a technology called AMOLED, and this AMOLEDs, they basically have, let's say, every single pixel has three different colors. So there you can really make a red, a green, and a blue portion of micro LED of OLED. And then this WOLED or white OLED is a bit the opposite. That's again creating a white OLED layer in general, and then using color filters to filter out the light. And what they did is they filter out red, green, and blue, but also a white part. So per pixel, you have red, green, blue, and white. So these two technologies were, of course, already an improvement compared to LCD. A big improvement, but it's kind of difficult to make, like, let's say, for example, the AMOLEDs on large scale and then the white OLED on smaller scale. Yeah, these kind of technologies were also not perfect. There were also, let's say, issues with brightness, for example. OLED, of course, had a good contrast, but then the brightness was far less. And this, of course, also needed to be solved. Then in came the quantum dot OLED. It's kind of, let's say, a marriage between those two OLED techniques that we see here. So instead of using a white layer of OLED, what is used there is a blue layer of OLED. So a blue emitting layer. And then quantum dots were used in the green and the red pixel. Let's say that the quantum dots are then inkjet printed on top of this blue emitting layer, and this is how it actually looks up close when you show a white image. So it's really interesting and nice to see. If you would want to watch a video that explains or that actually shows a breakdown of such a TV, so Peter Polomaki actually opened up such a TV and then really looked at the quantum dots inside, looked at how it was built. So this is definitely worth to do. To make this, let's say, this screen efficient enough, there were actually 3 layers of OLED used in the stack, and then with quantum dots on top, making a relatively efficient screen. There are quite a lot of applications, commercial products on the market, and this we see is really still on the rise. So it all, of course, started with a few monitors. The technology is all from let's say 2022, I think, and Samsung Display is really the let's say the lead in this whole story. And then the first let's say products that came out were from Samsung, Sony, Dell, and Alienware. But we see that it's really on the rise. Last year, there were gaming monitors, more TVs, more different type of monitors from different companies that all found their way to the market. So if you think about improvement of OLED with quantum dots, well, I mean, OLED already had a very nice contrast ratio. The manufacturability is a little bit less than LCD, for example. The energy efficiency is still, let's say, medium, especially because you still use a bit of filtering. The brightness was really a problem, and of course quantum dots don't really solve that. But of course, the colors improved a lot. So having a display with good colors and good contrast, this was of course the aim for the market. If you think about the requirements for these quantum dots in OLED, well, of course you need a high absorption. The photostability is already a little bit higher than what is needed for in LCDs. The thermal stability is quite low because there's not a lot of, let's say, light or energy that is dissipated in heat. And you, of course, need patterning. So this means that you need to go and inkjet print those quantum dots into the different pixels. Of course, this is still relatively low pixel per inch, so low resolution. So that's not really an issue for this specific application. Then we end up in microLED. MicroLED is a really nice technology, but of course it also has its issues. So maybe if you already have heard about OLED and LCD, maybe you haven't heard about microLED. Well, microLED is actually a technique where you use very small LEDs, so micron-sized LEDs. And then per pixel, you use a blue, a green, and a red LED. So of course, this image here is quite simplified. And let's say the size of these micro LEDs are here, it says between 3 and 20 micrometer, but we see in reality that people also tend to speak of micro LEDs when it's, let's say, 100 micron. So this technology is very new and very immature. So here the value chain is definitely still building up, and it's very exciting to see, because this is a technology that has, let's say, a lot of advantages compared to OLED and LCD, especially in certain markets. So first of all, for example, it can create very nice transparent screens, because the micro LEDs are so small compared to the actual pixel size. So this means that not a lot of space is taken up by the actual LEDs, and you can create a transparent screen very easily. Also, micro LED, because it's an inorganic LED, it actually has a very high brightness, and there's also a lot of design flexibility. So this is, let's say, ideal for cockpits or car displays. Also, when you think about wearables, you would want something that is very energy efficient, because of course you have a very small battery in such a wearable. And there, micro LED is also performing much better than OLED or LCD. And then if you think about these very, very large displays, the ones that you can find in a football stadium, at a concert, or in Times Square, for example, here you would want to have, let's say, seamless styling as well. So of course, all these displays are made up with smaller displays, so smaller tiles of a display. And then when you all bring them together, let them work together, that's how such a big screen is formed. But of course, sometimes you can see that it's the seam of these two specific tiles, so where the tiles meet. Because of the small fill factor that I already talked about, this is not the case for micro LED. So there are a lot of use cases, but today actually micro LED is still quite expensive. And this is because of the production cost. So when you would want to make a micro LED screen, you would need to start from a blue micro LED wafer, a green one, and a red one. And then you need to transfer all these LEDs specifically to the panel. And this is a very cumbersome and expensive process. So that's why it's actually useful to have color conversion there, so quantum dot color conversion, so that you can actually only start from the blue micro LED wafer and then transfer all the LEDs to a blue micro LED panel and then inkjet print. You might now wonder, like, okay, but why is that any different? Why is that, let's say, cheaper to produce in such a constellation? Well, it's because, first of all, the red micro LED is really expensive. It's about 20% more expensive than the greens and the red and the blues. So this means that if you can eliminate the blues, then you actually already reduce the costs a lot. Then the second part is the screen uniformity that would need to be improved. So the yield of such a display production, so the amount of displays that are usable after being produced, is actually also dependent on the variation of the LED wavelengths on such a micro LED wafer. And as you can see here, this kind of LED variation, it can vary quite a lot. And if you then make a screen out of that, you basically would have, let's say, different areas with different wavelengths. And of course, you will be able to see that. So either you only use a very particular part of your wafer and throw away the rest, which is very expensive, or you have a screen that you cannot really use. So this is extremely expensive, and that's why quantum dot color conversion can actually help. As Nico already mentioned, quantum dots can absorb a very broad range of wavelengths. So all of this can actually be absorbed by the quantum dots, which means that you can then make a very uniform screen in the end. So the yield improvement of this production process, of course, reduces the total production costs. Then on top of that, you also would need less repairs in the screen. So when you make such a screen, there's a lot of repairs to be made as well. And of course, you would need to replace every single LED. And this is actually helped today by using 4 LEDs per pixel. And then if one does not work, you just use the other 3. That might seem as something that is more expensive, but it actually is not. I'm going to hurry a bit because I think I'm running out of time already. So also the current density of the red LED minimizes the lifetime, and this is also a real issue. On top of that, you can make the micro LED displays much more energy efficient because you're starting from the blue micro LED, which is more efficient than, for example, the red one, which then increases your red subpixel efficiency. So that's also really nice. And this, in general, improves the micro LED technology so much. So first of all, you gain in color. The contrast ratio was already perfect. You gain a lot in manufacturability. The energy efficiency is also increased. The brightness might be a little bit of issue, but of course, the requirements for these quantum dots are quite stringent. So the absorption has to be very high. The photostability has to be quite high. There is thermal stability needed. The patterning could be low and high resolution, depending on the type of application that you would need. So this is still something where a lot of companies are actually working on, amongst our company, of course. But what I can already say is that there are today already quantum dot micro LED prototypes. Soflux, Foxconn and Applied Materials, Inolux, Raysolve, they all have been able to showcase prototypes on various fairs and industry events. So this is really coming close to being, let's say, commercialized in the market. And this is something where I'm really excited about, of course. We at Custom Dot, we actually make quantum dot inks specifically for micro LED applications. Our solvent-free ink is also processable in ambient conditions, which means that you don't need costly oxygen-free environments, which is very good. And then we combine that, of course, with the best-in-class optical properties. Then another technique that I briefly want to touch upon is the quantum dot electroluminescence, of course, because we have been talking about photoluminescence for a very long time. But you can also use your, let's say, quantum dots itself as nano LEDs. And these materials then can, let's say, can emit the light, but also form the light. So then you just need a cathode, the quantum dots, and an anode, and that's how your display is made. This is a really interesting technique, but of course, a very immature technology. The production yield is unknown. We all think that there might be a limit in brightness, but we don't really know. There's a question mark in the reliability of as well. Of course, the requirements are also a little bit different for these displays, but actually the first quantum dot electroluminescent prototypes, so the nano LED prototypes, have been already made. So at various events, you could have seen a prototype of Samsung, BOE, and Sharp. So maybe it's actually not that far away. And then in summary, so there are various display techniques that have been enhanced or will be enhanced by quantum dots. It all started in 2013 with the LCD, which also went through various, let's say, iterations. Then, of course, the QD OLED started in 2020. And hopefully, the first commercial quantum dot micro LED will come soon. And then, who knows, in the medium term, we will also have electroluminescent screens. So thank you very much for listening to this presentation. And of course, thank you, Johnny, for helping me with the presentation.
Thank you very much, Kim, for your presentation. So we are going with the last one. The best are always at the end, Peter. So that's your turn.
Thank you.
Peter will speak with us about quantum dots in NIR and SWIR photo detectors, opportunities and challenges. So without further ado, Peter, the stage is yours.
Okay, just a moment. I need to share the right screen. Just a moment. Yeah, I'm at minus 20 minutes, so I don't know if I can hold the time. Do you see my screen?
No.
You don't see?
No.
And now?
Not yet. If you want, you can always send to me the presentation and I will share it for you. And then you can tell me to change the slide when you need.
Just a moment. No, I will retry.
Okay.
Just a moment. You see something? You tell me when you see something.
Yeah, not yet.
Just a second. Okay, sorry for that. Yeah, we couldn't try it before because the connection didn't work before.
Yeah.
Just a moment, let me check.
Well, in the meantime, while Peter...
you can do the question and answers for the first two. Yeah, that's a good idea.
Yeah, we still do not have any question, but what I have to say to our audience is that a recording of this webinar will be available in the coming days. So please follow HortiQD project on LinkedIn, on Mastodon. We're also on Facebook, because the news to the website where the recording will be shared through social media. So please follow us and check it for the recording. Now we can see you sharing the screen, Peter.
Okay, that's good. That's already a good start. And now, let me see. You see now maybe the slides?
Yes.
And now we still need to get them on the... Is it okay?
Yes. Yes.
Okay, sorry for that. We'll try to get the laser pointer also. Okay. So we will switch gears now, because we had two presentations on the light emission with quantum dots, so for bio-applications and for LEDs. And now we will go to the light absorption. So we will go to photodetectors, and in particular in the infrared. So just two words where I'm coming from. So actually, I grew up not in the north of Germany like Nico, but really in the south, at nearby Karlsruhe, where I did my studies. And I grew up with bulky black and white cathodic TVs at that time. And then after my PhD, I had a chance to move to Grenoble, to SIA Grenoble, where I'm right now a research director in the fundamental research department. So Grenoble there's a lot of semiconductor industry, like with ST, for example, and there are even cities in the surroundings called Valence and Gap. So that's to show you how much semiconductor topics are going on here. So these applications you see right now here, they're all based on the use of infrared detection. So like in your smartphones, the facial recognition, or movement detectors, or night vision, or here to measure the oxygen level of your blood. And so this spectral range is very interesting for many applications. And here we will speak in particular about the sphere range. So sphere means the shortwave infrared region, and mostly located between 1 and 2.5 micrometers. And what's so special about it? So in this spectral range, many materials have a characteristic fingerprint, like plastics, food, minerals, or pharmaceutics. And it's also a spectral range which is less dangerous for the human eye. So if you have these detection systems, often you need also to send some light and measure it when it comes back. So you need some active illumination. And if you go to longer wavelengths, then you can use a higher illumination power without being dangerous for the eye. And so this gives you the possibility to have larger detection distances and higher precision. And so you have many applications, so like in quality control, in horticulture, in gesture recognition, in aerospace, and so on. And just to give you one use case, we can speak about plastic sorting. So this is an automated process. So the plastics in your waste are going on this band, and then they need to be sorted using a camera. And here, just to show you, when you go from the near range, so below 1 micrometer, to the sphere range, it will be much easier to distinguish between different plastics which are used widely. And this plastic recycling market is a strongly growing market. And of course, we want to reuse plastics, and when we want to decrease plastic waste. You all have heard about the problems in finding plastic everywhere on the poles, in the sea, and our bodies are already full with micro and nanoplastics. So we need to do something about it. And how does it work? So there you have this camera with optical system and some electronic processing, but in the core of this camera you have this sphere sensor. And the sensor itself, the image sensor, is composed of a material which is sensitive, of course, to the sphere range. And then you have also this readout integrated circuit which generates the electrical signal. And today's image sensors, generally in the sphere range, they use indium gallium arsenide, which is fabricated by epitaxial growth on indium phosphide and uses high temperature for the fabrication. And the problem with these devices is that the process has a low scalability, a low yield, and so you don't have that many units produced per year. And also the costs can be really high, from a couple of thousands of couple of 10,000s of euros per image sensor. And therefore you cannot find them everywhere right now. And this is where the quantum dots are coming. Of course, the idea would be to replace these infrared sensitive materials by a thin layer of quantum dots. And as we all know, quantum dots can be deposited from liquid phase on large area, for example, on the whole silicon wafer, and can be used in combination with well-established industrial processes. By doing so, of course, you can have much higher yield and you can strongly reduce the cost of your devices. So the advantages of the QD technology for sphere imaging can be summarized in this table. So here you have the established technologies like indium gallium arsenide or silicon germanium, which doesn't go that far in the sphere range. So the main advantage would be, of course, the cost reduction, the massive cost reduction by using quantum dots. And in addition, also the possibility to go to much larger areas of devices. And so the main advantages would be that we have processes which can easily be scaled up with low environmental impact. We can reduce the cost, and we are still compatible with the processes industrial used, in particular in silicon foundries. So I won't go into the details on the performance there. We can also already see, of course, the maturity is much lower than for the established technologies. The performances are a little bit lower, even though they're very much increasing right now. So here we have three examples of quantum dots. So lead sulfide, indium arsenide, indium antimonide. So why did I choose these materials? Because, of course, first of all, we need to have narrow bandgap semiconductors to start with. In quantum dots, as you all know, the bandgap will be increased by reducing the size. And so this means if you want to go in the sphere range, you have really to start with a very narrow band gap. And so not many materials are good for this range. So here you have a table showing this to you. And in the quantum dot materials mostly studied are lead sulfide, the 3,5s like indium arsenide, mercury telluride, and silver chalcogenides. There's already color code there because some of them are containing lead and mercury, and the others don't. And that has something to do with European regulations. So you have ROHS directive, which regulates the maximum amount of metals like cadmium, mercury, and lead in electronic devices. And so this can be quite stringent, and these regulations may change also. And therefore, today, I think many industrial players would like to get rid of lead and mercury and go to ROHS compliant materials. Nonetheless, lead sulfide is really the gold standard so far in this domain, in the near sphere photo detection. And the reason, there are several reasons, but an important one is that lead sulfide can be produced very easily as compared to other materials, and also it's less air sensitive. And so one way to produce these QDs is just by starting from substituted thiourea. You react them with lead eolate, and you get your particles. So that's very nice because you can control the reactivity of this sulfur precursor. And by doing so, you can have very good control of the size of the particles with really incredibly well-defined excitonic peaks. So that's most probably the best you can get with quantum dots, I would say, with lead sulfide, so in a very broad range, as you can see here. And there are other ways to produce them, and there are also commercial suppliers already today, like here, Quantum Solutions, or Work, or Nanoco, or I think even Custom Dots also, I heard recently, can provide lead sulfide quantum dots of very high quality. And there are also already commercial image sensors based on lead sulfide quantum dots. Here are two examples from Sphere Vision Systems. So one from 2018, this was the first one commercialized image sensor based on lead sulfide quantum dots, and then a much more recent one from last year. So that's actually a camera you can use like a normal camera to take photos in the sphere up to 2,100 nanometers, and with a good resolution. And so you can make even videos with it. So I don't know the price, however, so I cannot tell you, don't ask. And then also Emberion has launched, meanwhile, two lead sulfide-based QD imaging systems. So switching to ROHS compliant materials for near sphere photo detection. So 3,5s like indium arsenide or indium antimonide could be good materials. They are well known in semiconductor industry. They can be high quality and stable materials, but to produce them as quantum dots is really difficult, and it's a tough work because first of all, you have not a very nice choice of arsenide and antimonide precursors. They are more covalent, so you need harsh conditions, and they are very highly sensitive to oxygen and moisture. Nonetheless, you can do it, of course, and there have been several approaches. So today, most groups are using this kind of amino arsine or amino-stibine precursors because they're non-pyrophoric and at least sometimes commercially available, not all the time. And you can get rather reasonably well defined absorption spectra, which are indicative of narrow size distribution. Can do the same approaches for indium antimonide, which allows you to longer wavelengths generally than indium arsenide, which is often blocked at around 1.6 micron. But that's good. We have the quantum dots, but before making an image sensor out of them, we need, of course, to replace the initial surface ligands. Because when you synthesize quantum dots in organic media, they always contain long insulating organic molecules on their surface. And when you produce a thin film of these materials, then you will have very bad electronic transport properties. So you need to get rid of these long molecules and replace them by very short ones to improve the transport and at the same time passivate trap states. And this has been shown in this little bit older work here, where people check the influence of the spacer length between quantum dots on the mobility in a QD thin film. And you can see each time you reduce the distance between the QDs by 1 angstrom, you get 1 order of magnitude increase in mobility. That's not the whole story, because then already around 15 years ago, Dimitri Talapin and co-workers found out that we are not obliged to use actually organic molecules to replace the initial ligands, but we can use also inorganic complexes. So in that case, they showed it for Zintel ions like that one, the SN2 S6 4-, also called metal chalcogenide complexes. That was the first example. And then later they came others. So here, like with ammonium iodide, for example, you can see you can preserve the optical properties, and you do this biphasic ligand exchange, because what happens is that when you replace these long organic molecules with these short ions, you're switching from steric stabilization to electrostatic repulsion between the quantum dots. And I would say the backside of this approach is, of course, you will not be able to use conventional solvents like you would use, for example, for LED preparation, like octane or so. And you will need to go to polar solvents like DMF or butylamine, and which is more challenging to make nice films with those. And you can also dope quantum dots. So this can be done either by changing their stoichiometry, or by surface treatments. So once again, that's the example for lead sulfide quantum dots, where you can switch. You can see that the charge carrier density there, and you can switch from N-type to P-type just by changing the surface chemistry. And then if you go to the P-type materials, they're air stable then also. And that's very powerful. So you can make a PN junction with the same types of quantum dots. So today, there are mainly 2 types of colloidal quantum dot image sensors. And so the first one is the photodiode. So you have here typically the device architecture, where you have the quantum dot layers sandwiched between electron and hole transport materials, and then between the metal electrode and transparent electrode. So this device very much looks like a solar cell. So actually quantum dot solar cells, they have a very similar device structure. And the second one is the phototransistor, where in addition to the quantum dots, you have a thin layer of a 2D material like graphene underneath, which is a different device architecture, which can also give very good performances. So there have been some projections about the market growth of these sphere image sensors, and so Yole Intelligence, they made a very positive, very optimistic projection. This was in 2022 on this market growth. And just due to the fact that the established markets like machine vision and defense, they will be complemented by new markets, in particular consumer electronics, which is predicted to become a very important field for sphere image sensors. However, of course, such an evolution is only possible if the cost of sphere image sensors can be reduced drastically. And that's where I think industrials and research groups in this field think that quantum dots can play a very important role. So one example of this more consumer electronics is, of course, our HortiQD project. So just in few words, there, what we want to do is to check apples, so to detect apple diseases in an early stage by using hyperspectral imaging in the sphere. So you can see different apple diseases can have different spectral signatures in the sphere region. And so the idea would be that these automated modules are attached to tractors which are in the ore cart, and we will check the fluid quality. And the heart will be a quantum dot image sensor and also a Fabry Perot interferometer for this hyperspectral imaging part. And the outcome would be, of course, that we want to increase the yield and the quality of the fluid harvest, but also to optimize the treatment and to reduce the treatment, which can enhance sustainability and empower ecological farming. So with that, I will come to conclusions. So we believe that quantum dots can really lead to a paradigm change in sphere image sensors because we can produce large imagers at low cost. And in terms of materials, so 3,5 and silver chalcogenides can be good alternatives to established lead and mercury-based materials. However, there are many challenges still and a lot of development to be done. So we should integrate in our materials development really now considerations of sustainability, earth abundance, toxicity, and life cycle analysis. We need to enhance the stability of our quantum dots, in particular the thermal stability in the devices. And we need to narrow down the size distribution for the lead-free materials. And in terms of device development, we need to reduce the dark current in photodetectors. This can be done by improving the quality of the active layer, but also by optimizing the photodiode stack and the QD surface engineering, of course, which is really key for these developments. And these research directions, you can see it also there. That this is a very recent example from published by Sony in December 2024, where they have shown their data on indium arsenide-based, QD-based image sensors, so which gave very good EQE in the 940 range. And in this diagram, you can see also another example from Ted Sargent's group, where you can see also indium arsenide at 1100, and there they could really improve the EQE and reduce strongly the dark current by two factors, in my opinion. So first, the quantum dots look really monodispersed, so these spectra are really very good for indium arsenide-based quantum dots. And of course, looking at the energy diagram, we can see if you go to bigger particles, we cannot use the same stack as for lead sulfide. So we really need to know where the energy levels are and adapt the whole device architecture to make it work efficiently. And so this can strongly reduce the dark current in that case. So with that, I would like to thank my co-workers. So we have a fantastic team here working on this area, but also on other subjects. I would like to thank our funders, of course, and you for your kind attention.
All right. Thank you very much, Peter, for your presentation. So we have only one question. But this question is addressed to all the presenters by Dmitry Aldakov. So, but there are actually, there are coming other questions. So first question to everyone by Dmitry Aldakov is, do you think that the toxicity of the QDs due to the presence of heavy metals is considered as a real issue to be solved by the industrial players? Who wants to answer? First, Peter, you were talking.
It would be good also to have the answer of the others. So In the image sensors, I think it would strongly depend on the field of application. But of course, for if we talk about consumer electronics, it would be a very important point, even though you can generally stay below the limits imposed by the regulations, because actually you work with very thin layers of quantum dots, just 200 or 300 nanometers thick, and so you don't have that much in weight of the active material. But still, it's always good to say, okay, we don't have these toxic heavy metals like lead or cadmium. And then you have other sectors where I would say that that's not the main preoccupation. So it depends where. So if you have it in aerospace or in defense, for example, that's not the most important thing. So I don't know, but Kim, you also in this place probably there we have seen, of course, a big shift.
Yes, indeed, and I mean there are two things to take into account here. The first is definitely regulations by, for example, the European Commission. There is a thing called ROHS. So this is really important. So in ROHS, they actually restrict the use of cadmium, lead, mercury, and some other fluorine-based chemicals. So because of these regulations, there is, let's say, a limit to the use of cadmium and lead-based materials in commercial products. So this is of course the main driver of this kind of let's say shift in display from cadmium-based to indium phosphide-based, for example. But of course, then let's say the public image of such a company also plays a very big role. We see nowadays that the big corporations, even in China or in the US, they also don't use any cadmium or lead-based materials in their devices, just because it's bad for their image and for their sales.
Nico, do you want to add something to this?
Yeah, I think it's always, as I said, a problem. Maybe it's a philosophical problem, right? Because if there's no better materials, then you will use those materials. Very often, at least in diagnostics and clinical diagnostics where I work most, this is already a field where we try to heal people, right, or make a diagnostic. So it's with, whereas there's no problem if a pharmaceutical factory pumps all their stuff into a river that comes up, or when now, I mean, nuclear power was considered as dangerous some years ago, and you have to store things for millions of years safely. However, I don't know. But if there's a need, right, because now we all have AI that needs more energy, then people will make it happen. So I'm not saying that we should pollute more, so we should look for it. And if they're in displays, we should see where we, how we recycle those materials, same as for batteries before. I mean, the amount of cadmium that we use for an immunoassay or for sensing is so much less than the cadmium that used to be in batteries before. But I'm not trying to justify things with that things were worse before, right? So we should take care of it, but it's I mean, we cannot make lead or cadmium less toxic because it's just there. And if we cannot replace it, we probably have to use it. And as I said before, if we all don't want screens and live happily in the forest without polluting, then we don't need those things.
All right, thank you. So we have a couple of questions from Kishore Chiluka. So the first is, can 3V QDs ever match the optical properties shown by PBS QDs? and if there's any thoughts on how to improve the thermal stability of QD image sensors.
Okay, I think I will try to answer to that one. So what we see for the moment is that there has been a big improvement in the quality of 3.5 QDs these last 10 years. For the moment, even in terms of emission properties, so you can see now indium arsenide based QDs with decent quantum yields. In the size distribution is for the moment, I think it's challenging to get it down as much as for the lead sulfide, but it's doable. In the worst case, you can make size sorting, and you can get there. And what was the other one? Yeah, it was concerning the thermal stability. That's a very good question. That's a very good question. I think I don't have the answer on that, but it's for sure it's a problem, and it probably can be solved also by finding out the right surface treatments or shelling, or it's related to that, I would say. But that's an open question.
Thank you. We have one last question, always from Kishore. So, any thoughts on how to solve film cracking during ligand exchange process to improve the electronic properties conductivity of QDs in films?
No, you want to answer, Kim?
That's a good question. No, I don't know for the moment. One thing that I want to say is that we are not working on lead sulfide quantum dots, as you said, but more on indium arsenide quantum dots. We're not, let's say, that far that we already figured it out on how to solve that issue. Yeah, I am also very much looking forward to finding a solution to this.
All right. So if there's nothing to add to this, we can close our first webinar. So once again, I'm telling the audience that the audience will be able to find a recording of this webinar on the website and social media of HortiQD project. Please, you can also follow us to stay up to date for the next events that we will be holding. And that's it. Thank you very much. Thank you to our speakers, and we hope to see you again in the coming months for more interesting events like this one. So thank you very much. Goodbye.
Thank you for organizing, Claudio.
Bye. Thanks.
Bye.
Bye, everyone.
Thank you for participating.